All right, why don't we go ahead and get started? So the last time we talked about some of these hypersensitivity responses and then kind of finished up towards the end of the lecture talking a little bit, sort of a brief introduction about autoimmune disease. And I'm going to pick up more during today's lecture on that. And then, of course, you know, we won't be meeting on Friday because it's Thanksgiving holiday. So thanks to all you guys that showed up today. I guess some of you. So who is sticking around for Thanksgiving? So what are you guys going to eat? I mean, do they have like not McNuggets? What are they? Turkey McNuggets or something like that? Sorry, that's mean. Anybody traveling far away? Not far. Let's figure out who's the farthest away. Who's anybody going out of state? I guess those guys already left, right? So, all right. Anyways, so before we start, I just want to wish you guys a happy Thanksgiving and point out that we'll be back again on Monday, and we'll be talking about vaccine development and how the immune system is regulated by vaccines. And obviously, as you know, as the adult or the elderly person in this room, I have to, you know, admonish you guys to make sure to have a great time over Thanksgiving, but, you know, definitely want to see you all back here on Monday. So, you know, bright and early at noon. All right, so, so we're going to talk today. We spoke a little bit towards the end last time about basically what autoimmune disease is, and today we'll talk a little bit more about some of the specific autoimmune diseases. We'll talk about rheumatoid arthritis, lupus, type 1 diabetes. We spoke a bit towards the end about Graves' disease, and I'll just kind of briefly touch on that again today. And we'll spend some time talking about the mechanisms for immune tolerance and how that breakdown takes place that's required to develop an autoimmune disease. And then we'll talk about autoimmune disease development with respect to T and B cell collaboration. So you guys have learned a lot about, you know, for example, the germinal center response in lymph nodes, and how when that breaks down, or when that process takes place, you can develop autoreactive antibodies. And so you need to have both autoreactive T cells and B cells for that to take place. And we won't talk about celiac disease. We talked briefly about that the last time. All right, so what are the issues with autoimmune disease? So first off, when we think about autoimmunity, what we're really talking about is a hypersensitivity response like we've been talking about in the past. But in this case, this is not directed against an exogenous antigen. This is not an antigen from, say, pollen or bee venom or something like that. This is an antigen that is, you know, encoded genetically in our own bodies. And so the result of that is that if our immune system, for some reason, there's some sort of breach in tolerance, then certain cell types and certain organ systems are attacked by the immune system. And so we know that there are both autoantibodies as well as autoimmune T cells that have the capability of recognizing self-antigens, and when they do so, the results can lead to one of these autoimmune diseases. All right, so we briefly talked before, we've encountered immune tolerance mechanisms in the past, and as I mentioned, there's, you know, central tolerance and peripheral tolerance. Central tolerance, we call it that because it takes place in central or primary lymphoid tissues, and so for B cells, that would be in the bone marrow, and for T cells, that would be taking place inside the thymus, and it primarily involves this process of negative selection. So if there's a strong reaction between the antigen receptor, either on a T cell or a B cell, the result is that that lymphocyte dies and it doesn't get out into the peripheral immune system. As I mentioned, there's also antigen receptor revision, which is that if a B cell within the bone marrow, as it's undergoing this process of receptor rearrangement, if it generates an autoreactive receptor that then causes a signal to come back into that early B cell, the result is that it'll continue rearrangement until it gets rid of that autoreactive immunoglobulin molecule. If it doesn't, then it'll die. And then we also mentioned when we're talking about T cell development that there are these regulatory T cells. In this case, we call them natural or thymus-derived regulatory T cells because they're generated within the thymus, and they're basically have very high avidity to self-peptides and MHC. And instead of dying off during negative selection, instead they differentiate towards this unique population of regulatory cells. And we'll talk a little bit more today about how these regulatory cells work. So that's all the central tolerance. And then peripheral tolerance, we call it that because that's taking place in mature peripheral or secondary lymphoid tissues that we've talked about, like lymph node, spleen, and this gut-associated lymphoid tissue. And so some of the things that we've talked about before, and we'll come back to again today, is anergy. So if for example, if a T cell fails to recognize costimulatory molecules on the surface of a mature antigen-presenting cell like B7 through its CD28 receptor, the result is that that cell then becomes anergic, or it basically doesn't respond. So it doesn't subsequently respond if it's activated again. We haven't really talked too much about activation-induced cell death, but the idea here is that if a T cell is chronically stimulated, then it will die apoptotically. And then there's also this issue of clonal exhaustion. Again, we haven't really talked about it very much, but if say a viral infection, if you have some sort of a viral infection where the virus can't be eliminated, the result of that is that these T cells become clonally exhausted. And so they, sort of like anergic cells, they really don't want to respond at that point. And also there are a unique population of these regulatory cells that are not generated in the thymus like these natural regulatory T cells. And these are produced from naive T cells. So these T cells, in a sense, could become activated effector T cells, but instead differentiate into these regulatory T cells under the appropriate cytokines. And so the consequence of that is that they can block effector T cells from inducing an autoimmune response. And so we'll talk a little bit about how these Tregs work later. All right, and towards the end of the last lecture, I mentioned this Graves' disease, which is basically an attack inside of the thyroid gland. Specifically, antibodies are directed against this thyroid-stimulating hormone receptor. And the result of that is that, when we think about antibodies, sometimes we might think that the antibody can block something. It can block a cytokine, or it could block a receptor through steric hindrance. So it prevents this particular hormone, TSH, from binding the receptor. But in this case, these autoantibodies actually are binding and promoting the activation of the receptor. So they're basically mimicking what this TSH hormone is doing. And so the consequence of that is that you lose this normal regulation that exists between the thyroid and the pituitary. So normally, when the thyroid is activated, you get these thyroid hormones. They then go back into the pituitary and shut off this TSH hormone. The result of that is that you no longer generate any more of this thyroid hormone. But if you have a receptor here that's bound by this autoantibody, the result of that is that this is constitutively activated. So even in the absence of the pituitary's action on that receptor, you're constitutively expressing this thyroid hormone. And of course, the result of that is that you're what we call hyperthyroid, meaning that you have these high levels of these thyroid hormones in your body. And we know that this, obviously because it's an autoantibody, we need to have some T cells that are involved. And so there are Th2 cells that are autoreactive that are involved in promoting the generation of these autoreactive antibodies. So let's talk a little bit about how you might classify a disease as autoimmune. So as I mentioned before, when we say an autoimmune disease, what we're talking about is there's some sort of self-antigen that's being inappropriately recognized either by antibodies or by T cells, either CD4 or CD8 T cells. And in the case of Graves' disease, one can show that this is an autoimmune disease in a couple of different ways. The first of which is using an adoptive transfer model, where basically you take an individual that has Graves' disease, you take out some of the serum, and within that serum you're going to have some of these autoantibodies that can bind to that TSH receptor, leading to its constitutive activation. And then if you take those and you inject them into a healthy rat, there's enough cross-reactivity of this antibody for the rat's TSH receptor that it will induce a similar kind of disease in the rat, and the rat will then become hyperthyroid. So you can basically show that by taking this serum, that then will lead to a similar sort of disease in a different organism. That basically tells you that it's really something within the antibodies that's leading to this disease, and so that's how we can show that this is autoimmune-based. There are also experiments in nature, and an example of this, you guys have heard about the fact that mothers, there are antibodies that are produced within a mother that are received by the fetus transplacentally. So that normally protects the fetus during the first year of life or so because they don't really have a whole lot of their own antibodies at that point. But this can also be bad. So if the mother happens to have Graves' disease, making these antibodies against this TSH receptor, those can then also get into the fetus through this transplacental route. And the result of that then is that the child will also have this Graves' disease, at least for the first, until those transplacental antibodies start to disappear, which will happen within the first 6 months to a year. So how would you treat some, little baby that has this disease? One thing that you can do is you can remove the antibodies using something called plasmapheresis, which is basically where you take their blood and then you run it through a special machine that gets rid of all the antibodies that exist inside of their serum. And so the result of that is that because you've cleared off all those autoantibodies that are from the mother, these transplacental autoantibodies, that will then cure the disease, and this child will do fine. All right, so I wanted to talk about a couple of examples of autoimmune disease, and notably missing from this is multiple sclerosis. So I didn't have that in my slides, and I'm just going to point out that MS is a disease where you have T cells that are autoreactive, obviously, but in this case, they're reactive against certain proteins that are expressed in what's called myelin. So have you guys ever heard of myelin before? Anybody know what myelin does? Yeah. Yeah, so it helps in nerve conduction. So if you want to send a signal down a nerve, this myelin ensheathes the nerve and the axons in the nerve, and the result is that you get better transmission of some of these signals. So in MS, you have T cells that get into the brain where normally T cells don't go, and the consequence of that is that they start attacking these cells that produce the myelin sheath, called oligodendrocytes, and so then you can't make this appropriate level of myelin. And so with time, people start to become progressively more and more paralyzed, in fact, to the point where it can actually cause death. So that would be one example. That's a T-cell-based, CD4 T-cell-based autoimmune disease. Another example of this is type 1 diabetes or insulin-dependent diabetes mellitus. And this is an example, again, where you have a T-cell-dependent organ-specific autoimmune disease where T cells enter into the pancreas, and the result of that is that they attack certain cells called beta cells within what are called islets of Langerhans inside the pancreas. So this is the site where insulin is produced. And insulin is a hormone that controls glucose uptake in all of our cells in our body. And so the consequence of this is that if those T cells that are specific for antigens expressed on these beta cells, if those T cells get into the pancreas, become activated, then you have something that looks like this. So this is a normal, healthy pancreas. You can see the islet here. Over here, you have a slide basically showing a type 1 diabetes patient, a slice out of the pancreas. And you can see the islet here, but you can also see these little cells. Basically, this is something called a hematoxylin and eosin stain, or H&E stain. And it basically stains the nuclei in cells. And lymphocytes are very small, so they tend to show up as these little dots here. But basically, this shows you that you've got a lot of infiltrating lymphocytes inside of the pancreas that seem to exist around these islets. So the consequence of this then is that the islets become inflamed. There's a lot of inflammatory cytokines that are produced. And the islet cells, these beta cells, then start to get attacked. They get killed off to the point where you can't produce enough insulin to be able to deal with the body's needs for glucose. And so the result is that because you don't have enough insulin, glucose levels don't get taken up in the cells, and then you develop something called hyperglycemia, where you have very high levels of blood glucose. So people that have diabetes, whether it's type 1 or type 2, they have to measure their blood glucose in order to see whether they need to get an injection of insulin. All right, so I wanted to show a brief movie on this.
Diabetes affects the pancreas, an organ of the digestive system. An important function of the pancreas is to make hormones that regulate blood glucose levels. Insulin is produced by beta cells, which cluster in groups called islets. lowers blood glucose levels. After a meal, glucose reaches the pancreas via the bloodstream and is released from capillaries into the pancreatic tissue. This triggers the release of insulin, which is stored inside the beta cells. The insulin travels to other parts of the body via the bloodstream with three main destinations: muscle, fat, and liver. Insulin helps these tissues to store glucose as an energy source. Shown here are muscle cells. Insulin arrives through the fine capillary network that traverses the muscle fibers. The surfaces of muscle, fat, and liver cells are covered with insulin receptors. When these receptors bind insulin, they signal to the cells to release special glucose transport channels onto the cell surface. These channels enable glucose to gain entry into the cells, allowing the cells to store glucose as energy for future use. In this way, insulin acts to regulate blood glucose levels. In type 1 diabetes, a patient's own immune cells mistakenly attack the insulin-producing beta cells. Type 1 diabetes is referred to as an autoimmune disease because the body's immune system attacks its own cells in error. Over the course of the disease, a patient's immune cells will systematically and mistakenly kill nearly all of the beta cells, shrinking the island but leaving all other cells intact. The few beta cells that remain in the pancreas have a severely reduced capacity to produce insulin. The death of the pancreatic beta cells and the consequent inability to produce insulin is at the core of type 1 diabetes. Without insulin, the body cannot regulate blood glucose levels, and the high glucose levels damage many tissues throughout the body.
It's a dramatic soundtrack. All right. I just do that to wake you guys up. All right, so that's type 1 diabetes, and basically, as I mentioned, it's based on T cells that are reactive against these certain proteins that are produced by these beta cells in the pancreas. Now, another example of an autoimmune disease is something called lupus, systemic lupus, and it is basically, unlike the case for MS or type 1 diabetes, which are organ specific, in this case, it's more systemic. So you get these autoantibodies that are produced that circulate throughout the body, and then they can affect the function in various tissues, part of which has to do with generation of immune complexes, like we were talking about last time with respect to type 3 hypersensitivity responses. And an example of this is this sort of flushed skin on the face and other parts of the body that is due to release of a lot of these inflammatory mediators in the blood vessels within the skin. But this can also have an impact on a lot of other organ systems in the body, and it can also lead to death in rare cases. So what's going on with this is there are antigens that are recognized by antibodies, and typically these are things like nuclear proteins, DNA itself, chromatin, histones, and in some cases cell surface molecules. But they're typically, when somebody has lupus, you have these these anti-DNA antibodies that are recognized. And a lot of this has to do with, for example, defects in the complement system, because complement has an important role in clearing out apoptotic or dead cells from the body. If you don't do that very efficiently, the result is a cell can lyse, it can release some of its nuclear contents, and the antibodies are typically not going to recognize, they don't typically get tolerized to those intracellular antigens. And so the consequence is you can generate these autoreactive antibodies that can recognize some of these nuclear proteins. So I want to show a quick movie on that.
Normally, the body's immune system makes proteins called antibodies that protect it from illness. The antibodies attach themselves to harmful bacteria and viruses called antigens, which can cause illness.
Oops.
Lupus is a chronic autoimmune disease, which means the immune system mistakenly attacks healthy body cells and tissues without the presence of antigens. When this occurs, the antibodies are called autoantibodies. The result is inflammation and injury to cells and tissues that can lead to symptoms of lupus. There are different types of lupus. Discoid lupus is usually limited to the skin. Symptoms include a rash that appears as round, red patches on the face, scalp, trunk, or extremities. Usually, discoid lupus does not involve the body's internal organs, but the rash may be accompanied by joint aches. Systemic lupus erythematosus, SLE, can affect any organ or organ system, and most often results in episodes of inflammation in joints, tendons, and other connective tissues. Symptoms vary, such as fever, fatigue, weight loss, joint and muscle pain, swollen glands, hair loss, and nausea and vomiting. Because there is such a range in symptoms, the disease can often mimic other illnesses. The most characteristic symptom of SLE is a butterfly rash that appears in the cheeks and bridge of the nose. The symptoms can flare and remit over many years. The causes of lupus are unknown, although scientists believe that heredity and environmental factors are involved. This disorder is most common in women who are in their 20s and 30s. In some cases, lupus may be induced by certain medications. Lupus is diagnosed with blood tests and other diagnostic studies. It can be treated with medication according to the severity of the disease and symptoms.
All right, and one of the points in that was that in addition to attacking or to be giving that rise to this localized inflammation within the skin, there are some organ systems that can be affected by this, and an example of that is in the kidneys, where you get this immune complex deposition. Ultimately, that leads to breakdown in kidney function. So this can be pretty serious. The other point that was brought up in the video is that this generally tends to have a stronger impact in females. So you tend to find susceptibility higher in females, and that's generally true for a lot of these autoimmune diseases that we'll talk about. And it's one area of intense investigation, trying to understand why females, particularly in the 20s and 30s, are more susceptible to things like lupus and diabetes and MS. Now, another example of an autoimmune disease that you guys have probably heard about is rheumatoid arthritis. And in this case, you can see this, there's a lot of inflammation going on. And if this is chronic over time, you can see the sort of devastating impact that this can have on the joints. And basically what's happening here is that you have both T cells and antibodies that are autoreactive. They go into the joints, and in particular, they target what are called synovial joints, where you have these articulating bones, and there's a layer of fluid within this synovial cavity that basically cushions and lubricates that joint that allows you to move. So if you have an autoimmune response that's taking place here, the result of that is that this gets inflamed. You got a lot of these inflammatory cytokines and edema and so forth, and the result of that is that this movement can be painful and ultimately can be quite debilitating. So some ways that you can treat this, one of which that's been quite effective over the last 15 years or so, is to block TNF. So we've talked about TNF in the past when we were talking about, for example, type 1, Th1 type cytokine responses. So TNF is pro-inflammatory, And it's known that there's a very high level of TNF that's expressed in these arthritic joints. So if you block this, so there's a medicine called Remicade, there's a couple of other pharmaceutically available biologics also that target the TNF pathway. And basically what these do is they block that inflammatory process, and the result is that it reduces the inflammation. But it doesn't cure the underlying autoimmune disease. So you still have the autoreactive T cells, you still have the autoreactive antibodies, and so the consequence is that you can treat the symptoms, but you really don't develop a cure against that. But we do know that it's effective. So for example, if you look at some RA patients, and if you treat them with a anti-TNF antibodies to block the TNF pathway, you can see that it has a really strong impact on pain, on the swollen joints, as well as the release of something called C-reactive protein, which is part of the complement pathway it's actually released by the liver in response to systemic inflammation. So basically all of those parameters go down if you treat with anti-TNF, and that has a nice effect on trying to treat the symptoms of this. Physicians can also use anti-inflammatories like non-steroidal AIDs and so forth. They can also use, to try to get rid of the autoantibodies, they can also try to deplete those with a molecule called anti-CD20. So this is a molecule that binds to, or an antibody that binds to CD20, which is expressed on B cells. So by depleting these B cells, then you can reduce the production of these autoantibodies, and that will also have not a curative effect, but it will reduce the symptoms of this sort of disease. All right. So let's talk about some of the factors that are involved in autoimmunity. So we've heard in a couple of these videos about what's happening in terms of breaking tolerance, right? So you need to have both tolerance in terms of the antibodies as well as tolerance in the T cells broken somehow, and the result of that is that you can now generate this autoimmune response. But I think it's important to note that the question is, why does this happen? Why does certain people become more susceptible to certain autoimmune diseases versus others? And we know that there's, at least there's a lot of reasons for this. It's multifactorial, partly because there's a genetic predisposition. So as we'll talk about, in particular, there are certain haplotypes of MHC that will predispose you to particular autoimmune diseases. And we also know that there are environmental influences. And so the combination of the two, these are typically not single gene defects. They're usually the consequence of both having this genetic predisposition as well as environmental influences. And that could be microbial infection. It could be cigarette smoking, a whole variety of different factors that are involved in that process. Now, an important thing that I want to point out is that, a lot of these autoimmune diseases, we talked about some T cell specific organ-specific autoimmune diseases. We also talked about some where there's an antibody involved. And even with that, the key is breaking T cell tolerance. So B cells, as you guys think back when Dr. Fruman was talking about B cell development in the bone marrow, those B cells, if they recognize self, they can either do this receptor editing or then they'll die off. But there's not a system like the AIR system for T cells, which gives rise to expression of antigens in the periphery for the B cell system. So they don't recognize all the potential tissue-specific or they don't have the ability to become tolerant against all the antigens in the body. So all of us have circulating autoantibodies, but the important point is that we don't have autoreactive T cells, or not enough of them to really predispose us to a particular autoimmune disease. So, and this is true not just for the T cell mediated autoimmune diseases like MS and rheumatoid arthritis. That's also true for antibody mediated autoimmune disease like Graves and SLE. So the question is, why is that? And the reason for that is that, as I just mentioned, the B cell tolerance is not as tight, it's not as stringent as you have for the T cells. And you need to have the T cells there in order for the B cells to undergo this affinity maturation and isotype switching. So in order for autoantibodies to be effective, they have to be high affinity, just like they're effective for getting rid of a pathogen or something like that. And so they require T cell help for that process. All right, so why is that? So let's talk a bit about, go back to our discussion about T cells inside of germinal centers and the follicle. in a lymph node. So here you have this high endothelial venule, you have B cells that are coming in, and these are entering in through this T cell zone, and they're going to try to make their way to the follicle. In the follicle, there are these follicular dendritic cells, which are required for those B cells to stay alive. So they produce tonic factors to keep those B cells alive. So this is an autoreactive B cell. It comes into this T cell zone and starts to recognize self-antigen here. And so the consequence of that is that, just as a B cell would getting activated over here, it wants to stay in this T cell zone because it wants to set up these interactions with T cells to get the signal to undergo somatic hypermutation, isotype switch, and so forth. But because of the fact that it's stuck here and there are no T cells in this T cell zone that are specific for that particular autoantigen, the consequence is that these B cells then die off. So you're not going to produce this high affinity autoantibody because the B cells, once they enter into this T cell zone and there's no T cells there that recognize that autoantigen, they're going to undergo apoptosis and die off. All right, so that would be if these autoreactive B cells are coming in through the blood, but that also happens within the germinal center itself. So here, let's say that you have a B cell that's undergoing somatic hypermutation, right? So the point of that is that when you have a B cell interacting with a T cell, they can undergo this process of refining their antigen receptor to try to make higher and higher affinity antibodies. And the process of this is essentially random. So you get these mutations that take place within the B cell receptor genes that can promote either lower or higher affinity or whatever. But on top of that, the consequence of these mutations is that some of those B cells might develop autoreactive receptors at that point. And we're no longer in the bone marrow here. We're not talking about B cell development, we're talking about mature B cells. And so if they make this mutation such that the B cell receptor is now overtly autoreactive, the consequence of that is that there are not going to be any T cells around that can provide help anymore because you don't have these autoreactive T cells, or you don't have many of them in a normal individual. And so the consequence of that then is that the B cell, after making this switch to this autoreactive antibody, that B cell is then going to die off apoptotically because it doesn't get T cell help to allow it to continue along. Any questions about autoantibodies? Okay. So now in terms of the T cell tolerance, I've been sort of harping on this idea that the T cell tolerance is really, primary in this, that you need to have, all these different mechanisms in place to keep these T cells from, inducing an autoimmune response. And one of the examples that we talked about before when we were talking about T cell activation has to do with this concept of anergy. All right, so this idea that normally we would think that most of the autoreactive cells would be removed in the thymus during development by negative selection, but there's always going to be some cells that get out that are sort of weakly autoreactive, and they escape. So how do we prevent those cells, once they get into the periphery, from becoming activated against our own tissues? So this comes back to this issue of co-stimulation. So here you have a T cell, and we've talked about this concept that you need to have two signals, one of which comes through the T cell receptor binding to MHC plus peptide on the surface of an antigen-presenting cell. And the second signal, which is this co-stimulatory signal, comes through, in this case, CD28 expressed on the surface of the T cell, binding to B7 on the surface of this antigen-presenting cell. And remember that for this antigen-presenting cell to be what we call a mature APC, it needs to have seen some sort of, molecular pattern associated with infection or with some sort of cellular damage, right? So that results in activation of this antigen-presenting cell. It upregulates B7 on the surface, and then the result of that is that when this T cell recognizes this antigen on this professional antigen-presenting cell, then that gives both of the signals, then the T cell is productively activated. If the T cell interacts with an antigen-presenting cell, and let's say that this is a self-antigen, and in fact, most of the time, the peptides that are going to be presented by MHC are going to be self, okay? They're going to be coming from within or near from the outside of that cell. But in this case, because this is an autoantigen, it's not associated with some sort of an infection or something like that. The result of that is that there is no B7 on the surface of this APC, and so you just get signal 1. You do not get the co-stimulatory signal, and so the consequence of this is that the T cell becomes anergic. It is functionally non-responsive. And of course, if you just get the second signal, if there's no antigen that's recognized by the T cell receptor, that second antigen, or that co-stimulatory signal alone will not activate the T cell. So you need to have both of these signals in order for the T cell to become activated. And this really ensures that this response, this activation of this T cell is only taking place when there's something bad happening, whether it's a microbe or there's some sort of a, what we would call a damage-associated molecular pattern. Okay, so that's an important point that controls peripheral tolerance. On the other hand, certain autoimmune diseases like MS, for example, have been associated with viral infections. And so, what might be going on there is that you may have some antigen-presenting cells that, because of a viral infection, they may have a weakly autoreactive T cell receptor that can recognize self-antigens. And because of the fact that now you have some infection going on somewhere, you have mature antigen-presenting cells that can present those peptides. And so the consequence then is that you get both of these signals. You break tolerance, and even if it's sort of a weak avidity interaction, it's still enough to activate that T cell because you now have as well this co-stimulatory signal. So the consequence then is you start to generate an autoimmune response. So this would be one example of an environmental factor that helps to promote autoimmune susceptibility. We've also talked about regulatory T cells, and we talked about that in the context of both these natural regulatory cells that develop in the thymus, as well as these induced regulatory cells that can develop from the naive T cell pool in the periphery. And they are essentially CD4 T cells, so they're basically like helper T cells, and they're characterized by the fact that they have the expression of something called CD25, which is the high affinity receptor for the IL-2. And as well, they also have this molecule called CTLA-4. So CTLA-4 is normally associated with non-responsiveness in T cells, but in this case of these regulatory cells, their role is to suppress other T cells. And in this case, we can see a CD4 T cell on the surface of an antigen-presenting cell, and this autoreactive regulatory T cell can suppress this conventional T cell that wants to become an effector T cell and essentially keep it from promoting an immune response. And it's part of there's probably about 20 different mechanisms for suppression about how this all works. But some examples of this involve things like IL-4 and IL-10, which are anti-inflammatory cytokines. So by expressing these anti-inflammatory cytokines, these regulatory T cells can basically block these conventional T cells from becoming activated. And one thing that allows us to recognize these cells, as well as something that's crucially involved in both the differentiation and function of these regulatory cells, is a transcription factor called FOXP3. So these regulatory cells have this high level of expression of this FOXP3, and the result of that is that leads to the generation and function of these regulatory cells. There is an example of where this is deleted or it's defective in certain patients, and that leads to an autoimmune disease called APE-CED. This is a broadly systemic autoimmune disease. If you don't have these regulatory T cells, the result is that you've got autoimmune disease all over the place. Any questions about regulatory T cells? All right. So I mentioned that there are certain environmental factors that are associated with autoimmune susceptibility, like virus infection, that sort of thing. But I also mentioned that there are genetic factors that are involved. And certainly one example of a genetic problem would be if you had this mutation in FOXP3 that's involved in generating regulatory cells, and you develop this systemic autoimmune disease. But other examples are associated with HLA itself. So if you think about it, HLA, because of the fact that there are a lot of different haplotypes out there, certain individuals are going to be more susceptible to autoimmune diseases than others, simply based on their HLA, and I'll explain why that is in a minute. And this is just an example. If you looked at the different haplotypes in a population, we have all sorts of different haplotypes out there, but in siblings, they're going to have basically, just through random assortment, just Mendelian genetics, you're going to get roughly the number of shared haplotypes, between, let's say, 4 siblings. Some of them are going to have no shared haplotypes. Some of them are going to have both paternal and maternal haplotypes, and then some will have one or the other. And so, again, just standard Mendelian genetics here. If you look at in folks that have autoimmune disease, and this is an example for diabetes, what you see is that you don't have this Mendelian ratio anymore. It doesn't appear to be just random assortment, because now you're selecting on the basis of having this disease susceptibility. So what you see is that folks that have one or two alleles that are shared, two haplotypes that are shared, tend to have a much, I should say, the folks that have those shared haplotypes typically have diabetes. So the consequence, basically what this tells us is that having those shared haplotypes doesn't necessarily mean that you're going to get the disease, but it's a susceptibility, enhances your susceptibility to that particular disease. So what do I mean by that? So this is just showing some of these different susceptibility alleles for all of these different types of autoimmune diseases. Ankylosing spondylitis. Here you can see that a particular haplotype for B27, so this is a class 1 haplotype, is observed in greater than 95% of the patients. So having that particular haplotype for class 1 gives you a roughly 150-fold increased risk than the normal population. So obviously that's a very, very strong disease association. And then you can just go down the list. And some of these are class 1 alleles, some of these like for narcolepsy and celiac disease, type 1 diabetes, et cetera. Those are class 2 alleles. But the important point is that you can see that the disease risk is much greater. So this kind of connection between the MHC haplotype and disease susceptibility is quite high in some of these. And it tends to go down. So some of these, maybe they have a lower disease susceptibility allele as you go. And in fact, there are actually some where there's a negative correlation. So in this case here, having DQ6, this class 2 haplotype, makes you much less susceptible than somebody that has either DQ8 or DQ2, which have a 14-fold higher susceptibility. So you can see how there's this association between MHC and disease susceptibility. So why would that be? What's the basis for that increased risk? So this is an example of how your particular MHC haplotypes could potentially influence the kind of T cells and the kind of antigens that can be presented. So you've heard before when Dr. Fruman was talking about these different HLA haplotypes that exist out there, that some of them are better at presenting certain peptides than others. And obviously, most of the peptides that are going to be presented are self-peptides. Either they're going to come from inside the cell and get on MHC class 1, or they can come from the outside of the cell and get on class 2. But the important point is that you're going to have tons and tons of self-antigens presented. Some of those antigens may be better at promoting a particular type of T cell response against self. And so this is just an example where you can look at some of the amino acids that are present here in this DRB subtype, this class 2 subtype, that raises susceptibility to certain autoimmune diseases. And it's really just a matter of just how well certain autopeptides, these self-peptides, are presented that enhances the susceptibility to certain autoimmune diseases. Okay, so it's sort of a complex issue, and it's still not very well understood how this all works. But we definitely know that there's an association between some of these alleles and others. And as I mentioned, it basically suggests that certain autoantigens that might exist, for example, in pancreatic beta cells, there's some autoantigen there that if you have a particular MHC allele, particular haplotype, you get much better expression of that particular peptide. And the consequence of that then is that you have T cells there that can recognize that and cause disease. So any questions about this HLA connection with autoimmune disease? All right. So the last thing I want to point out is that there are additional features that can promote autoimmune disease. So we've talked about MHC susceptibility alleles. We've talked about infection and so forth. But another example of this is physical trauma. So the body has certain places that we call immune privilege sites, and one example of that is the brain. Normally you don't want to have T cells go into the brain and start attacking tissue. Why do you think that is? Why would you not want to have T cells surveying the brain all the time and getting rid of stuff? Yeah, so the neurons are not particularly good at being regenerated. So, you know, as we'll learn over the weekend when we drink a lot of beer and stuff, we'll lose a lot of those, and then anyways. So, no, but the point is that it doesn't do a very good job. The neurons don't do a very good job at regenerating. And so the immune system really tries to avoid that, because the immune system is really good at killing off things. And so it doesn't make a lot of sense, you know, to send T cells up there, which is not always the case. I mean, there are certain examples where, you know, if you get an infection in the brain, you do get T cells, you do get NK cells and other inflammatory cells that can get in the brain and try to get rid of that. But oftentimes there's a consequence to that. You know, you have the immune system attacking some of the neurons, and that can lead to things like MS, for example, or you know, what happens with West Nile virus infection. So another example of this sort of immune privilege site is within our eyeballs. So our eyes are a place where T cells don't normally go, lymphocytes don't normally enter into that, and there are cells there that basically prevent those kinds of responses. But if let's say that you get in an argument with somebody at a bar and they punch you in the eye, you know, normally you get a black eye and it's no big deal. But there are situations where the consequence of that is that you can start to release, for example, these intraocular proteins, which are, again, normally sequestered from the immune system. But if you damage some of the barrier function within the eye, those antigens can then leak out and make their way to a nearby draining lymph node, be presented to T cells, which presumably there are enough of those cells that are, you know, sort of weakly autoreactive that you can get a response. And then the consequence is that the T cells come back out of the lymph node, and then they go to the eye where it's, you know, where you have a black eye. It's been punched. And so this actually can be so serious that it can lead to blindness. Now the issue that's problematic is that, you know, now because it's not just the damaged eye, you also get recruitment of these T cells into the normal non-damaged eye because of the fact that now these T cells can recognize an antigen that's present within the eye. And so the consequence of that is that this can then lead to blindness. All right. So, so any questions about physical trauma or this particular response? All right. Happy Thanksgiving. We'll talk again on Monday about vaccination.